Okay, so before we jump into this particular question, we need to take our time around what it is that this is asking. So let's break it down. So in a monohybrid cross, so mono meaning single, hybrid, if the P generation consists of plants that are true breeding. And so look, this is a term that we don't use a great deal in our lectures. So hence the reason why I've indicated here homozygous. True breeding just means that if we have a plant that has a particular trait, for example, might produce 2 leaves, it doesn't matter how many generations we look at, each of those generations will produce the same phenotype. So hence that wording, true breeding. Really what we're saying here is that they are homozygous. So if we go back to our round alleles, here we would have a homozygous situation. And it's only those alleles that are going through. And so essentially what we're saying is that, okay, so here we have a single hybrid cross. If the P generation consists of plants that are true breeding, homozygous, then the F generation will consist of organisms that exhibit what ratio of dominant phenotypes to recessive phenotypes. Now, this question requires us to understand that we have 2 plants involved, one that has a dominant phenotype and the other one that has a recessive. So let's have a look here. So if I've got a true breeding plant that is RR, and I'll draw it in black, and then I've got another one here in red. Okay, so both of these are homozygous. We know that because the question says it's true breeding, and I've indicated that it's homozygous. If we do the cross, what we find is that, let's set up the Punnett square. So the 2 alleles from the red plant are small r, and then here we've got large R, capital R, capital R. Okay, we set up our Punnett squares here, and so we end up with the dominant alleles being present in all of the offspring, as well as the recessive. However, the question here is asking about phenotypes, not genotypes. And all of these plants here, because they have a single copy of that dominant allele, will all express the dominant phenotype. Hence the reason why E is the example. None of these plants here, none of these plants will exhibit the recessive phenotype. So this is our F1 generation. Okay. Next question. In a monohybrid cross, if the P generation consists of plants that are true breeding, then the F2 generation will consist of organisms that exhibit what ratio of dominant phenotypes to recessive phenotypes. Essentially, this is Mendel's work. So if we come back here, where we've got one plant that has this, and another plant that has that. So we've got our dominant shown here in the capital letters. Sometimes we will refer to it as being plus as well. If we cross these plants, so this is our P generation, the F1 we saw previously will generate all of the plants will have this genotype. Now if we back cross or allow these hybrids to mate with themselves, we end up with this situation where we have 2 different alleles. So we have a single copy of the dominant and the recessive. Okay, so here we've got a homozygous dominant allele. This one will be heterozygous for both the dominant and the recessive allele, and then the final one will bring together those recessive alleles. Now, in terms of the phenotype, we have at least one copy of that dominant allele. And so therefore we will have 3 plants exhibiting the dominant phenotype for every 1 plant that has a recessive phenotype. So hence the reason why D is the answer here. If it had asked about genotypes, that would be very different. Okay. Here what we would find is that 25% would be homozygous dominant, 50% would be heterozygote, and then finally 25% would be homozygous for the recessive. However, the question asks about phenotypes, and hence we have this 3-to-1 ratio. As I said, this is essentially all of the Mendel crosses that I showed in class. Okay, so coming to Mendel, so if we have a look at his work, the principle of dominance states that in a heterozygote, one allele may conceal the presence of another allele, and we just saw that previously, where if we have this is our dominant allele, this is our recessive allele, the presence of that single dominant allele will mean we will have the dominant phenotype being expressed or observed. Okay, so next question. If an allele has the same phenotypic effect in heterozygotes as in homozygotes, that is genotypes AA and AA, are phenotypically indistinguishable, then the allele is dominant. So it's just another way of saying that essentially the allele has the same phenotypic effect, be it single copy or double copy, 2 copies. Okay, so dominant. Okay, so here we're looking at this issue of codominance, and the example that I gave in class was about blood types. So I'll just bring up a new slide here. In terms of the blood types, so if we look at the phenotype, okay, so we have blood group A, blood group B, and blood group O. And essentially what we're looking at is, if I draw a cell, a red blood cell down here, what we're talking about are the presence of these surface proteins. Okay, so I'll just draw a line here. So it could be that we have protein A being produced, and that would give us this phenotype here, blood group A. Alternatively, we can have a situation where we have blood group B being expressed. That would give us blood group B. Where we have no protein being expressed on any part of the cell, this gives us blood group O. Okay, so for us, and hence the reason why O, blood group O, is actually a recessive phenotype. We rarely see blood group O, and that's because we only require the presence of one allele to produce A or B or O. The final blood group that we have is AB. And here we can have a situation where we have both protein A and protein B being expressed on the surface of that red blood cell. So in terms of the genotypes then, we can have homozygotes for A and also for blood group B. We can also have a situation where we have AO and BO. So here we have a single allele, single copy of the A allele, or we have a single copy of the B allele. But because this allele is dominant to O, we still have the blood group A or B. The only time that we get blood group O is when we have homozygotes for the O allele. And then in the case of the blood group AB, we have both an A and a B allele. So we have one copy of the A allele, one copy of the B allele. So in this system, we actually have 3 alleles. Okay, so a lot of our examples prior to this, we're only looking at 2 alleles, a dominant and a recessive. In this case, we have 2 dominant alleles. Oops. That are codominant, which means you only require we only require a single copy of those alleles for us to observe that phenotype, and the presence of one does not negate the other. We do, however, have one recessive allele. The only time that you'll see its expression is when we have homozygote form of it. Okay. If this is still a little unclear, feel free to email me or have a read through the texts as well. Okay, so moving on from Mendel's work. So really, the focus of Mendel's work was about showing us this idea that genes are a unit of inheritance, that genes are independently assorting. So they're transmitted through the paternal line or the maternal lines, independent of each other. The genes are being transmitted independent of each other, and that we've also got this idea of dominance and recessive. And so building on these concepts, we then started to looking at the relationships between family members. And the tool that we use to do that is a pedigree chart. Okay, so